Well, welcome everybody. It looks like we filled the house today. Just in case it's helpful, there are 2 seats in the front, and I see maybe there's a little bit more room on the stair there somewhere. And though there's one here, and we can take a minute or two for people who would like to sit. I see a chair, 2 chairs right here. Come on down. I know there's some students and postdocs up there who want to have a seat. We have 2 more down here. Okay, I see. Yeah, come on. It's okay. All right, excellent. Really, I am really excited to welcome you here today for our 2023 Daniel I.C. Wang Lecture on the Frontiers of Biotechnology. As our tradition, I'm going to tell you a little bit about the namesake of this lecture series, Professor Daniel I.C. Wang, who received his bachelor's degree and his master's at MIT in 1959 and 1961. And he got his PhD in chemical engineering from UPenn in 1963. Then Professor Wang spent 2 years in the U.S. Army, and he was working at that time at the U.S. Army Biological Laboratories at Fort Detrick, Maryland, from 1963 to 1965. And he joined MIT faculty in 1965. So here we have a young Professor Wang. Professor Wang founded the Biotechnology Process Engineering Center through the NSF Engineering Research Center Initiative. And he was its director from 1985 to 1998, which is a significant amount of time for a center of this nature. That center was really quite foundational in establishing biotechnology as a field and training many in the field and establishing it. He also was the part of the summer short course that was the longest running and most successful, also based on biotechnology. So Danny founded that short course series. And he was subsequently named in his later part of career as an institute professor at MIT in 1996. In 2000, Professor Wang was given the Temasek Professorship at the National University of Singapore. And there's always been a strong Singapore connection with Danny. And he devoted part of his time to help Singapore in their biomedical science research and development. Professor Wang received many awards, including the ACS Marvin Johnson Award and David Perlman Memorial Lecturer. He also received awards from the AICHE, the Food and Pharmaceutical Bioengineering Award. He was the AICHE Institute Lecturer, and he won the Walker Award, which is one of the most significant awards that you can receive in the chemical engineering field. He was given the Amgen Award in Biochemical Engineering from the Engineering Foundation. He also received the Asia Pacific Biochemical Engineering Award. Professor Wang has been elected to the American Academy of Arts and Sciences, the National Academy of Engineering, the American Institute of Medical and Biological Engineering, Academia Sinica, which is the honor society in the Republic of China, and the International Institute of Biotechnology. He consulted for many companies during his time, including 50 companies worldwide, and served on many boards. He also co-authored 5 books and over 250 publications and 15 patents. So just a little bit more about Professor Wang. I mentioned his time in the service. And of course, very important to him were the people that he trained. And we have many examples of that. Here he is standing with some former students, and Professor Cooney is one of those. There are many in the room as well. Now, I want to thank Nubarafan, the sponsor of our DIC Wang Lecture. He's our very own alumnus. Dr. Nubarafan is founder and CEO of Flagship Pioneering, a company that creates bioplatform companies to transform human health and sustainability. An entrepreneur and biochemical engineer, Dr. Fan holds more than 100 patents and has co-founded more than 70 life science and technology startups during his 36-year career. He is co-founder and chairman of the board of Moderna, the pioneering messenger RNA company. Noubar entered biotechnology during its emergence as an academic field and industry, completing his PhD in biochemical engineering at MIT in 1987. He was a senior lecturer at MIT Sloan School of Management from 2000 to 2016, a lecturer at Harvard Business School until 2020, and he currently serves as a member of our MIT Corporation. He teaches and speaks around the world on topics ranging from entrepreneurship, innovation, and economic development to biological engineering, new medicines, and renewable energy. In 2022, Noubar was elected to the National Academy of Engineering. Dr. Fayon has received multiple awards for his passionate advocacy of the contributions of immigrants to economic and scientific progress. He is the co-founder of the Aurora Prize for Awakening Humanity and a number of other philanthropic projects. Now, I am very excited to introduce our DIC1 speaker today, Mr. Stéphane Bancel. Stéphane Bancel has served as Moderna's Chief Executive Officer since October 2011 and is a member of Moderna's Board of Directors since March 2011. Before joining the company, Mr. Bancel served for 5 years as Chief Executive Officer of the French diagnostics company bioMérieux. I hope I pronounced that well. Thank you. From July 2000 to March 2006, he served in various roles at Eli Lilly and Company, including as Managing Director in Belgium and as Executive Director, Global Manufacturing Strategy and Supply Chain. Prior to Lilly, Mr. Bancel served as Asia Pacific Sales and Marketing Director for bioMérieux. Mr. Bancel currently serves on the Board of Directors of Indigo. He is a venture partner at Flagship Pioneering. Mr. Bancel holds a Master's of Engineering degree from École Centrale Paris, a Master of Science in Chemical Engineering from the University of Minnesota, and an MBA from Harvard Business School. So I am very excited to introduce our speaker, Mr. Bancel.
Thank you so much, Paula, for the kind words. Good afternoon, everybody. It's quite impressive that so many of you came on a Friday afternoon to be part of this lecture, so thank you so much. As you can imagine, it's a very humbling moment for me to be here today because of the very nature of this lecture. Madame Wang, thank you so much for being here. Just think about the impact that Danny had. Noubar trained with Danny, like many. Wei Shu Hu trained with Danny. Wei Shu is the one who trained me in Minnesota. Danny had no idea when he trained Noubar and Wei Shu, and then Wei Shu trained me, that all of us and many others together we're going to build Moderna. It's just one example of the amazing legacy that Danny had. So in his memory, thank you so much for everything that he has done and to his family. So usually for most CEOs, it's pretty stressful to think you're going to present where 2 of your board members are in a room. Because the way to think about board members is like bosses. So I have 8 bosses, which is always fun also. That's another thing that people don't think about when they want to be CEO. They don't realize that it's come with a package. But the great news is Bob and Noubar are not only on the board, they are friends, they are my partners, and have been my partners since the very, very early days when Moderna was just an idea in our heads. And we're working together to try to figure out how do we make this happen. So I need to go back a bit in a time machine with you, because of course you know some of the answers, which is yes, mRNA works. But in 2010, 2011, when we were putting the company together, when Bob and Noubar were talking to me about this idea of using mRNA so that humans will make protein in their own bodies, they will turn humans as biotech factories. For all of us, chemical engineering, that was a very seductive but very kind of crazy concept. And so the first time I remember vividly in the winter, Q1, I think, of 2011, when Noubar is the first person who mentioned to me this idea. As you can imagine, my first reaction was, you're crazy, right? You're going to inject mRNA in people, and the mRNA is going to be stable, it won't be immunogenic, and it's going to make the right protein on demand. But Nubar was very, very good at not trying to brute force the idea through my throat and my brain, but to help me think about it. And he asked a very simple, very powerful question, which is, what if mRNA can work safely in human? And with this, he kind of flipped me like a crepe. And I started playing the game with him. Well, if mRNA works safely in human, you're going to be able to make a lot of amazing, innovative medicine for patients. A lot of them. Like in the hundreds, maybe over time, talking decades, maybe thousands. Because if you think about the biology of cells, you could, of course, get an mRNA in cells. And once it's in the cell, the ribosome just doesn't think. It just reads the message and make the protein. And then through signal peptide, you can tell the cells to do what you want with the protein. Get it out of the cells and get secreted. So you can do what biotech can do. You can tell the cell, put that protein you just made on the cell membrane, which you cannot do using recombinant. You can do cocktails of mRNAs, because in every one of your cells, you have a lot of mRNA at any moment of time. And so if nature can do it, the only question from engineers was, how do we understand how nature does it so we can do it in a factory and in a vial? And so we thought about doing very complex proteins. We have now an mRNA program in phase 3 that has 6 mRNA molecules in the vial. To do biology that was just unthinkable to do using recombinant. And then you can also go and access compartment of a cell. We have right now in pivotal study in rare genetic disease, a protein that is made in the cells and then is carried into the mitochondria, because it's a protein deficiency inside the mitochondria. And you can just think about all the things you can start doing and think about over time all the things you could do. Because we are all very lucky. We live at a time of explosion of medical knowledge. There's not a week that goes by with an amazing new paper coming out where we collectively understand more about how the human body works. But with such a technology, because mRNA is information, you could very quickly translate that into a medicine, like the team has been able to do with COVID. As we've said in the media, 48 hours to design in silico the vaccine. The true story is it took the team 10 minutes. They literally on the internet copied and pasted the spike protein sequence. They went into our drug design studio. They copied, pasted it. They pulled down menus for the lipid we wanted, the manufacturing process. In 10 minutes, they were done. And then I freaked them out. Because they're like, we're done. And I'm like, are you sure? And I'm like, what do you mean? I'm like, this is going to be in a human in 60 days. And they're like, we don't have time to do mouse work and in vitro work. Like, guys, he's going to be in a human in 60 days. Are you sure it's the right one? And you see people turning white, yellow, green, say, oh, geez. I said, take a day or two if you have to, because that molecule has to be correct. And so they went back, spent the whole weekend. It was literally a weekend. Spent the whole weekend looking at literature of the previous experiments, protein, 3D structure, and so on. And literally by Sunday night, Monday morning, they're like, yeah, we're sure. With the full length, not fragment, full length of spike. I said, OK, here we go. They click order. He went to a factory in Norwood, and 60 days after, he was starting his first clinical trial in Seattle. So the ability to do a lot of things that you could not even contemplate to do using recombinant or small molecule. The other piece that was extremely interesting is we believed that this technology was going to have a very high probability of technical success. As you know, and I will talk about it in a minute, in pharma, 90% of the drugs that start clinical studies fail. That is not a good return of R&D investment in the pharmaceutical industry. But if you think about it again as a chemical engineer, and you think about the molecules that we inject in people, it's always the same. The 4 nucleotide just change the order, will code for something else, but the same 4 chemical matters that from product 1, product 2, product 50 you put into your human body. And then the lipid you put around it is also the same for every application. And if you think about it, most drugs in pharma today do not die because the drug don't work. they die because they are toxic or because they are toxic at the dose where it works and you have no therapeutic index. And so that notion that we'll use for every product the same chemistry from a safety standpoint was extremely powerful. The third piece was speed. As Noubar knows, I'm really, really patient. And one of the things I was so frustrated in pharma was how everything takes so long. You go to meetings and people talk in years, and every little project is in many, many quarters. And when you're not patient, it's not so fun. And so the piece that was very obvious to us, again, as engineers from the beginning, which is if we could invest a lot in process engineering and in robotics and in digital, you could go very, very fast because every time it's the same manufacturing process. Unlike in my olden days, as I'd shown as a chemical engineers in recombinants, where you have to develop a new process, new cell line, optimize everything for every drug. And sometimes they are really hard to make. But what if, like we've shown in COVID, you can just go and it's the same as last time? And last but not least, having been more on the business side of the house for a long time, whereas Noubar kept being entrepreneurs and being very close to the science, I had totally forgotten how you make mRNA, and I asked him, how do you make mRNA? And he literally answered to me, I don't know if you remember those words. He said, as a ChemE, you're going to get the kick out of it because it's liquid phase, enzymatic reaction. And I replied to him, oh shit, it's going to be really cheap. And then I paused because I'm like, you tell me that if this thing works, that I think is not going to work. We're going to be to do hundreds or thousands of drugs as an industry over time, that the chance of going from the lab into approval is much higher, potentially the inverse of what you see in pharma, 10% success. Could you have a world where it's 10% failures? And you could go very fast in the lab, very fast in the clinic, and they could be at scale affordable. And so I paused. I'm like, what am I missing? It just seems too good to be true. And that's basically how my journey into Moderna started. So as we discussed, the drug discovery and development, the pharma, biopharma, is a very bad business, if you think about it as a business person. And there's a lot of studies by a lot of big consulting firms showing the return on capital of the industry is going down like a straight line year over year. Because it's really, really hard when most things don't work and they take a lot of time. That's really, really hard. But if you think about it, doing a startup in biopharma, which is why I think Noubar is totally crazy, is even more crazy. Because you have to do with those challenges, the odds are against you, it takes a lot of time, and then you have no infrastructure, no money. That's what he does for a living. That's what Bob does for a living when he starts a new company, right? And so that's a piece that you have to put your head around about thinking about science-based businesses, is you really trying to go against the odds. So as I've reflected over the years about one of the reasons I think Moderna was successful is we were able to run this cycle many, many times. And I think this is basically a central piece of how you build a biotech company, my humble opinion. It needs to be science-based. And we started with a little bit of science ideas and investigation. You need to raise some capital. You need to get some people to move the science further, to raise more money, to get more people, and to keep going. The key is to keep things moving. If you don't have scientific progress, it's not going to go very well. Because next time you go asking for more money, you might not find a lot of people. And so the key is, how do you think about risk? How do you pivot quickly? How do you obsess about learning fast? Because time is your enemy. What I think a lot of entrepreneurs do not realize in any industry, but especially in this industry, where it's really hard because it's not an engineering world, it's mostly a science world, is how time is your worst enemy. Time is your worst enemy. And so thinking about what you do, what you don't do, in which order you do things, how as you get new data, you pivot very quickly. Sometimes we reorganize Moderna on one set of new data. And I mean, literally learning something new that was unknowable before us, because there was no book about how to build an mRNA company at the MIT bookstore, at the Harvard bookstore. And so sometimes what we did with the team is we literally learned a new set of facts when we were sure about the fact and the data was correct. we reorganized the whole company, meaning on the board, we drove 30 people and jobs at the company and go sleep on it at night, come back in the morning, are we sure? And then go into the lab, get the team, explain the data and the science, and explain the new organization and say, any questions? Okay, go back to the lab, we're changing, and we're going 90 degrees right or 90 degrees left. And that's what you need to have the courage to do that time and time and time again. And sometimes it's really tiring. But that's what you have to do because time is your biggest enemy. And facts and data are your biggest friend. But you need to have the courage to do the right thing, and sometimes it's not easy. What I like to joke about with people is that the capital intensity of this business is so high. I tell people, if you had told me in March 2011 when Nubar and Bob convinced me that I should quit my nice, comfy CEO job and start again and help the team and be part of the team building Moderna, that I will have to raise $5 billion, I will 100% chance have not taken the job. And that's what we ended up doing with the team. But if you had told me you're going to have to raise $5 billion, are you ready? I would have said no. Say first, because it has never happened before us. There's no biotech company before us that has raised $5 billion. And I never raised a penny in my life. I think about it. At Lilly, I never raised money. We're having cash flow coming out of wazoo every day. And bioMérieux, same thing. Never had to go and convince people to open their checkbook based on your idea. Sometimes we raise big rounds, like at the time, $500 million was in biotech very big round. Preclinical, meaning zero clinical data. That was a very big round. And because we will not publish at the beginning to protect the science and file the IP and the patents, sometimes people in the media will mock us, saying they must have very nice slides because they can raise a lot of money, but we have seen nothing. So that's kind of an important piece of a biotech company. So as we said, we must have been kind of crazy because we went into the biopharma R&D world in a startup and in a new technology. So if you think about just compounding risk, that's kind of, I think, the top of a mountain in terms of compounding risk. Go work in an industry that is capital intensive, where most things don't work. Start from nothing, and then do it in something where nobody knows how to do it. And so Nubar got a very nice analogy for us in the early days. It's like you need to be paranoid like you're climbing Mount Everest for the first time, with no GPS and no map because nobody has ever climbed it before you. That's a mental model you need to have, and that has been such a useful tip. To be paranoid while optimistic, to question every decision. Like if you pick the wrong turn on the wrong path, you're going to die. And if you die, but you believe that there's a chance that somebody in the world could make mRNA work, there's a lot of drugs that are going to come that won't happen. And that will be on us. So there's a lot of intensity about finding the right path. New technology, which was a challenge in itself. Again, go into a time machine. I'm running around Kendall Square in Cambridge, $2 million in the bank. That's what we started with, trying to hire people. What are you doing? mRNA. How much money do you have? 6 months, maybe 9 months. So you want me to quit my job to come to work on this crazy thing, and in 6, 9 months we might be bankrupt? Yeah. And so it will be either finding people in academia who will be good at mRNA, but I have no idea how to do drug development, because that's not what you learn in academia. You learn science. Or people from the industry who could barely know how to spell mRNA. That was one of the other dilemma we had in terms of execution, is how do you build that team to start? Because you need to start climbing the mountain. But you could die in the first few legs with the early team. Which is why I was very, very lucky to be able to have a board and a scientific advisory board that were very, very hands-on. I see sometimes some entrepreneurs, some CEOs, keeping the board and everybody away. And my strategy was exactly 180-degree opposite. Get as many people as you can under the tent because you want to make the best decision you can. Because you cannot make the wrong decision because you have ego in the way. You have to get the best mind around the table. So many times we had breakfast at 7 o'clock in the morning where we bring some bagels and so on. We would crash the call the night before just to look at data and to decide what do we do. Because that is not what we expected. Good data you can inform by a quick email. But bad data, you need to figure out what do you do next. So that was really, really important in terms of our challenge to start the company. An amazing part about building Moderna was it was very clear to all of us that from a scientific standpoint, this could not be a 1 or 2 drug company. It made no scientific sense. Because of the information nature of mRNA, it will be either 0, because we will go bankrupt before we have the cash coming through the door, or it will be a lot. And when I mean a lot, I mean a lot, a lot. So that was super helpful to us. We were lucky from a strategic standpoint, because I spent no time about 1 or 2. I spent no time about 0. So it was all about how do you build, but how do you manage risk? So if any of you are curious, spend 5 minutes on the internet, go Google the Moderna S1. That's the document you file when you go public. And while like any S1, you will find 60 pages of legal jargon by the lawyers about all the risks that could happen. Because we're so obsessed about risk management, if you look at the upfront section, written in plain English by business people, not by lawyers, we articulated 4 risks that were central to us and that we talked about all the time. With a board, with a management team, every time. If you be a fly on the wall at Moderna, you will hear us talking about those risks many times each day. And one of the central risks we had, in addition to financing risk, we need to raise a lot of money over time, and if you can't, then that could kill you. Execution risk, biology risk. There was a huge technology risk, which is nobody knew where this thing was going to work best first. And we were very conscious that if we pick the wrong place first, we might kill the enterprise, because if the first thing fails, it's not going to look good. We were all a bit frightened by what happened to gene therapy. Some of you are way too young, but some of you remember what happened in the 90s with gene therapy when the first clinical study at Penn failed, with a kid dying. The whole industry was put on ice by the FDA. No more funding to understand what happened. So a lot of work happened then in academia, but in industry, the industry was over for a long time. And so this was also always in the back of our head, which is we cannot trip on the first clinical study. That would be really, really bad outcome. And so what we try to do and think a lot about is try to assess from a technology standpoint where were the risk of all the things you could do. Because one of the beauty of mRNA and Moderna, one of the challenges of mRNA and Moderna, is you could do almost anything you want. Which protein you want to make? Just tell me. We go on the internet, get the sequence, make it, and a few weeks after, we inject the mice, and you have a protein in their blood. So which drug do you pick? Which biology? Why? And then which application? Do you go and start in the brain? Or do you start as something that looks mundane, like a vaccine? And as you know, our first clinical study that we started in December 2015 was in vaccine. Because as we talked with the board and the team about all the things we could do, what we all aligned on is that most probably the least risky thing to do, again, there was no book to know that from, but just team talking and challenging each other, was don't use a human protein. Because if you make a human protein and there's an antibody response to the human protein, you might create an autoimmune disease into a healthy person, because I think you don't know, and that was unknowable before in animals. Vaccine is low dose because the immune system amplifies the instruction from the antigen. It's 1 or 2 doses, not a rare disease where you need to dose a kid every week or every 2 weeks forever. So there's a lot of features about vaccine that seem to us, again, being paranoid about we don't know what we don't know, so we have to be so paranoid and careful. But the vaccine space seems to us most probably a wise place to start the first clinical experiment. The other thing we did, and Ubar has a lot of credit for it, is we tried a lot of things in preclinical model first. We didn't rush to the clinic because we wanted to learn and build optionality. We could have picked, like a lot of companies do, pick one thing and rush to a clinic because it's going to be easier to raise money when you're in the clinic, which is true for most companies. But what if your rush to a clinic kills the company? Because you take a big detour in a mountain and you end up in a very bad place where it's really, really cold and it takes you a long time to come back onto the right path. So So there was a lot of discussion on the technology risk, and then a lot of discussions on the biology risk. There was an interesting moment in Moderna history when we were picking, okay, what antigen do we code in the first vaccine? And I remember some of the team members saying to go and speculate about which product will have a big NPV, you know. And I told them, I don't care about the NPV of the first product. And the scientific team looked at me like, do you went to Harvard Business School? I said, yeah, I did. But I learned also math in elementary school, which is $5 trillion of NPV times 0% chance of success equals $0. So I would rather do the other thing, which is 100% chance of success on the biology side of the house times $0 equals still $0, but I would have learned something and I would have succeeded somewhere. So we say we need to find something that we will know if it worked or not. Because if we go to the clinic the first time and the clinical study fails, you will have no idea if your technology was not ready or if you had the wrong biology. And how can you learn and improve on something you don't understand? You can't. And so we're obsessed about going step by step in the journey, which most people thought we were crazy. Again, if you go back to the media, the good and the bad thing about the internet is it's still out there. So people say they are crazy. They're going after a vaccine, which is a crappy business, and they're doing a vaccine for which there is no market. Those people are really like crooks. They're crazy. And we did it, I hope, in a very thoughtful way, which was it was not about making the first product the biggest. It was about building this platform. This was his obsession about it will be zero or a lot. Don't try to be too smart about the first one. If you really believe, and we're all super aligned, which is what was amazing about Moderna when we built it. A lot of people who understand science deeply and believe in science and facts, not in what has been done by other people. So we said in the early days, we're going to throw the biotech playbook by the window because it doesn't apply to us. We're building a platform that is information molecule-based, and we have to invent and sometimes make mistakes and be comfortable with our mistakes to learn and to invent how we do it. So technology risk, don't focus too early, but explore. Partner with companies. Again, it goes back to this financing, this credibility, validation for financing. We did a very large partnership with AstraZeneca in March 2013. I believe at the time it was the biggest biotech deal for a preclinical company ever. $240 million upfront. So remember, I was refreshing my, at the time, Silicon Valley Bank. Don't worry, we have other banks now. But Silicon Valley Bank, like all day long, because we had at the time around, I don't know, $10 million, $20 million in the bank. So trust me, it was nice in one of the refresh all day long when it went from like $20 million to 260. So, oh, wow. It's not the same company anymore. And one of the things that Nubar did to help us say, we need to reinvent the strategy of a company. Because it's not the same company anymore. It cannot be the same strategy if you have $20 million in the bank or $260 million in the bank. So we spent the next few months after, on the one hand, building and hiring to execute the commitment we made to AstraZeneca, because we made a lot of commitments to get that much money. But we knew we could make it happen, and we did. And in parallel, it was, okay, what does it mean for the company strategy now? And hire people that have seen the movie to help us scale and de-risk the company. I think that has been fascinating about Moderna, and I was a bit slow in the early days. I think it took me a good year or a year and a half to understand that we were in an S-curve. I had seen those many times in business book, but again, I was slow. It took me a while. One morning, running on the Charles River, I'm like, geez, we're in an S-curve. And the reason it happened is that, the chemistry team will come with a new nucleotide chemistry, and it will be 5x better. At Lilly or at bioMérieux, 5% was a good year, but 5x. But then literally 2 or 4 weeks after, the formulation team will come. Oh, we have 7x better. I'm like, you mean including the chemistry improvement? No. On top of the chemistry improvement. And he kept going and going and going. And after a few times it hit me, I processed and I'm like, geez, what's going on? And it's like we were just, after a while where we were not moving, because there were months like it was really, really painful, not moving, like nothing was working. But we are learning. We are learning a lot. And we're obsessed about the learning. And then we started taking off. And that was really, really interesting. And so when this became very clear, we say this is going to be a 20-year game. And the key is to be the company who's going to learn the fastest. And Stephen Hawking, who runs the science, had this motto, say we don't have to be the smartest, because he will be arrogant to think we're the smartest, but we absolutely have to learn the fastest. And so to learn fast, we basically understood at the time all the components of the mRNA platform we were building, and we built massive robotics so that questioning science was not rate limiter. That if we need to do 500 mRNA in a month, we could do 500 mRNA in a month, to do as much as we can, to just go and explore the space, to learn as fast as we could. And so we built that very early on in the company history. Now it's a huge, huge operation in Norwood. And for us, that was super, super important to be able to scale very quickly. Another piece that I thought was really important as we built the company was to be clear on priority. And the slide could have been better. It's on me. We always say priority number one is quality. If you do crappy science and you have crappy data, well, good luck. Because you do a business that injects people in human, you have a very strong moral obligation, an ethical obligation, and by the way, legal obligation, it's called FDA, to do things extremely well, and quality cannot be compromised. Like when you do airplanes, if you put people in an airplane, you better make sure quality is there. Then we're obsessed about speed, so that was number one. Number two, speed. Then scale. How do we scale? If you think it's going to be tens and thousands and hundreds of drugs over time, you have to figure out how to scale. And cost was priority number four. You have to set priorities because you set the culture. You know, the Walmart culture is cost, always lowest price. It works for them extremely well and for the customers. But when you build a business, you have to be very clear who you are and who you are not. And as part of leadership, you have to be very clear and repeat and repeat and repeat so people do it the way you expect it. You don't have chances if you are not clear because everybody's going to do it their own way because that's what they think you want. And the biggest thing of responsibility of leadership is to be very clear where are the priorities because people have to make trade-off decisions all day long. And if you don't tell them a guidebook, they're going to pick it up. And you might not be happy with the outcome. So it's really, really important to set very clear priorities. Another long discussion we had over many, many years is going public. Being private was wonderful. You say what you want, when you want, to who you want. I think too often biotech companies see going public as an end game. Going public is a means to an end. It's a financing event. It's a one-time event. It's not a goal in itself. It's a means to building a business. But it comes with a lot of things attached, i.e. being public, where you have massive disclosure obligation, you have a lot of pressure. The day you go public, as I like to kid CEOs that I'm coaching, is the last day you pick the stock price. Because when you're private, you negotiate with a set of buyers the stock price. And I tell people, the day you set up with your board the price of the IPO is the last day in your life you're going to set up a stock price. From the next day, it's a lot of crazy people who set up the stock price for you. And the piece where I think I was really lucky is I see a lot of biotech CEOs that think that it's a big badge of honor to have taken a company public. But I was lucky because I ran a public company for 5 years before Moderna. So taking the company public was never a badge of honor for me. It was not an end game. It was a means to an end at the right time. And we've always said, and we always debated a lot, which is very important and very healthy, is when do we go public? When does it serve the mission of a company? When does it serve the science? And I'm so thankful to our investors and to our board to have allowed us to stay private so long. We went public in December 2018, which I think yesterday I got an email, was the 5-year anniversary of us being public. And we went public because I failed raising more private money. In January 2018, we'll raise our last private round, and I told the board, after everybody was very nice with nice congratulations word, to say, we need to go public now, because there is no more money out there in the private market. We're done. So we have to go public because we need more money to fuel the science and to fuel the engine. And so we need to go public now. Hey, just a few departing thoughts because I'm much more interested in your questions. A few things. You know, I like to say, you know, in business school, they teach you real estate is location, location, location. Well, business is people, people, people. I've been lucky because, you know, I grew up in my career at Lilly, which was still is, I believe, I'm not there, but I believe a very people-driven culture. But building Moderna took that to yet another level for me. As Nubar and Bob know, when we started, there was nothing. When I resigned from bioMérieux and I arrived at Flagship on a Monday morning in July 2011, I had a laptop, a phone, no meeting in my agenda, nothing. And that was literally Moderna, a bit of money in the bank account, a couple of people who had been hiring as I was being recruited. And it's people who built everything. The science, people. The drugs in the clinic, people. The factory in Norwood, people. The IT system, people. It didn't fall from the sky. People, blood and sweat to build this. And so as I like to tell people, if you want to build an amazing business, if you do it to average people, you have no chance. You need to hire amazing people, you need to develop them, you need to grow them, you need to make sure they're in the right roles, you need to coach them. But you need to have a very, very high bar, which is hard. Remember what I said at the beginning. Nobody wanted to come work with us because it was crazy and we had no money. So how do you thread the needle for that, and how do you upgrade the team? How do you move people around? How do you reinvent, rebuild the company all the time? The other piece that I learned being an entrepreneur is you have to be fearless. As I like to tell people, you have to be totally relentless. Like if a door is closed, you have to go by the window or by the chimney or by the basement. For some things, you have to go through a brick wall. The key thing is that to be ethical, it has to be legal. Those are the boundaries that are uncrossable. But in those boundaries, you have to figure out what is the thing you have to do to get the job done. And I was so pleased during COVID, a little anecdote about the importance of culture. During COVID, you know, engineers designed a lot of machines, scale up everything very, very fast. And one day what happened is a new machine that we designed, built at a company was delivered, and nobody had the time or the insight to check if it will fit through the door. So it gets delivered into the warehouse, and then it's on wheels, it doesn't fit in the door. Because it was a madness, as you can imagine, during COVID. We worked 7 days a week. It was just crazy. And so you have a bunch of engineers from big pharma that we hired to think, what do we do, and a little meeting. And there's a guy called Ben who has been with us since we were 20 people. So Ben knows the Moderna culture inside out, backward, and forward. What do you think Ben did? He went to a workshop. He literally took a sledgehammer. He came back, and he took the wall down. And as the pharma people were discussing what should we do, he was wheeling the machine, said, please call facilities, tell them we have to rebuild the wall. And he was going, and he pushed the machine. So we gave him the price of a quarter. We celebrate our values of individual or teams. And I tell that story at every onboarding that I do every month when we have new team members joining. Because Ben knew this is what we would expect of him that was right for his family, is the patients. I always tell people, use your family as a mental model for thinking about patients. So it's not an abstract concept, but a concept very close to your heart. And he knew that if he would ask us, should I take the wall down? I'd say yes. But he knew that in this culture, doing a risk-reward analysis that was on mission, legal and ethical, that was the right thing for patients, he would have our backing. And he didn't realize he was going to be celebrated at every town hall. But that's really important, again, the role of leadership to set the culture. Because it's easy we make the right decision when I'm in a room, but that's not success. Success is the team make the right decision when I am not in a room. That's success of scale, and that's culture, that's mindset, that's the people you hire. We talked about learning. You guys are all engineers, I assume, or most of you, or all of you. I think one of the most powerful law of physics is 1 + x to the power n, compounding. And if you can find a way to compound on very, very quick cycles, nobody can catch up with you. So you have to decide for your own life, for the businesses, the science, the labs you're going to be running, how quickly are you going to compound, you and your team? Stephen Hogue, who runs our science, His compounding time is 1 day. When he was running the science and the platform for a long time, he will meet with his team every day at the same time in front of a wall. They will draw on the wall, and he will ask everybody, what did you learn since yesterday? What did you learn since yesterday? And he wanted everybody to hear it. And they will debate the ideas, and they will reprioritize in real time based on the positive and negative information they learned in the lab every day. So we're compounding the science every day. That's how you learn really fast. Okay? I've been super lucky because when I see entrepreneurs that start like Nuba, building a company out of grad school or college, I'm like, geez, I went to business school, I went to a big company, I was even a CEO, and it was hard. These last 12 years, it was hard. Even with the pandemic was really hard. But even forget the pandemic, it was hard. It was so hard to build a business from scratch, a science business. And the last thing is, choose your partner wisely. I've been so blessed. My wife is my best friend. She has been so supportive in everything I've done, including deciding to join these guys to build this company, and then to invest all our savings into the company. She's like, now you're very crazy. And through COVID and through everything, who you become over time through your life is based on the people you surround yourself with at work, in your home. And so very important to pick your friends and your partners wisely. So with this, I'll be delighted to take any question you have. Thank you.
All right, excellent. We're going to start with questions, and we have plenty of time. This is wonderful, and I will help to move the microphone around. And we have a microphone help back here, so raise your hands, and we will get questions. Here's one right here.
And so you talked about how the scientists would meet every day and talk about the things they learned the previous day. I feel like some days when I'm working in the lab, I don't learn anything. I just do the same things, and it worked, and so I kept doing it. Do you have any examples of mundane things that people would learn in one day that they could share, even if it wasn't an amazing discovery?
Yeah, so not everybody was learning new things every day, to your point. But because he was running a cross-functional team where he had chemists and process engineers and biologists and tox people, different people will learn something each day. And what he wanted first is the team to learn everything as a system. Because the drug that's in the vial, you have to make hundreds of decisions about the drug that you design in the vial. And so he wanted to first train people to think about all the pieces and the trade-off and the tensions. Because in most places, people optimize for their piece. But at the end of the day, you need to optimize the system. What might be good for the drug might be really hard for the chemist, or for the tox people, or for process engineers. And so not everybody had things to contribute every day, but they were all learning every day. And what they will do, when somebody will learn something positive or negative, they will reestablish, is the science we are doing, are the experiments we are doing worthwhile, or should we stop doing some of them? The other thing that he will do is quite interesting. In most places I've seen before, people hate failure. In the previous setup I've been in, when there's a failure, everybody goes away. Like nobody was part of that work. And when something doesn't work at Moderna, Stephen gets really excited. Like he's almost like a kid in a candy store. And the first few times I was like, what's wrong with you, dude? And it's like, we're going to learn so much. Because it's not black magic. We thought it was going to work. It doesn't work. We're going to learn a lot. And that's why he will go in hyperdrive in front of his board and then use the team that I would have been learning over the last months and quarters and years. Say, OK, we thought that was going to work. It didn't work. Why? And he will just go and listen to every idea, regardless of people's levels, how long they have been in the building, every idea, put the idea on the walls. And then when the idea list was exhausted, he would have a group critique every idea. So do we need to do the experiment, or it's a bad idea? And then sometime he'll come in my office, and I will dread those days, because he'll come. This doesn't work. I'm like, shit. He's like, don't worry, we got it. Those are the 5 things we're going to do. By the way, I need $5 million. I'm like, what? Because he would go crazy about doing a lot of experiments at the same time. And sometimes we say, this I need to do in monkeys. I could kill you, and we're going to do it in mice and rats and feel good. We need to do it in monkeys. But I need like 20 monkeys or 50 monkeys. Oh, shit. But that's the judgment that you need to make the decisions to be excited about failure, and to have prepared the team for that moment. So yes, you're right, not everybody learns every day, but if you work as a team with people from other field of expertise of science, then you go learn new things most days.
Thank you.
Thank you.
Thank you for the talk. Very inspirational. You did talk a lot about the beginning, the risk that you had to take, how crazy that idea was. But I want to discuss risk. Did risk gradually decrease as you grew? Or is risk right now still a huge factor in what you do at Moderna?
It's a great question. So the risk, so first, two things. I said 4 risks, huh? Technology risk, biology risk, execution risk, financing risk. First, one of the crazy things we did, and I think it was the right thing to do, is because we are so paranoid about technology risk and the unknown and biology risk that exists in our field, we build a portfolio like you do with stock. If you know which company is going to go 100x in the next 2 years, you buy only one stock. But because nobody knows that, what people do in financial industry, they build a portfolio to manage risk. So we did the same thing. We ended up having very quickly 16 drugs that will all prosecute at the same time. But what we did by doing that, we increased tremendously execution risk. You have to do all that at the same time. So it's much harder to do. It's exponentially harder every time you add one. And we took the financing risk through the roof. Because you don't need like $50 million, you need $500 million a year. So every year we'll go and have to raise $500 million or the plan could not be executed. So over time, risk was titrated. As we learn more in the clinic, of course, technology risk decrease. Because the good thing about mRNA being information is if one vaccine works, we know a lot of vaccines are going to work, like a lot. And so we then double down, triple down, more vaccines going to the clinic because that risk was off the table. There's still risk in the other application that we are playing with. But it could allow us, by reducing technology risk, then to double down on that application. The financing risk, of course, when we became profitable, has gone away, in a sense. We don't rely on other people's willingness to give us money to fuel the company because we become sustainable from our own cash generation. If you look moving forward today, I would say the biggest risk we have today is an execution risk because of the complexity of what we are doing. You know, we have plans to launch 15 products in the next 5 years. AstraZeneca, which is a company I think 20 times our size, is planning, which is a great company. Huh? I think they're planning to launch 17 products in the next 7 years. And we don't have the infrastructure that such a company has. So we are building the airplane as we are flying it. So that's the risk we have now. Is I literally have a mental model of people like Ben on the wings, just putting the engine, on the wings and make sure it's really tied well to the wings. That's what Moderna feels some days. And so the risk we have today is really an execution risk, and what I call a culture dilution risk, which is, again, you guys are chemical engineers, so it's fun to talk about that. I think about a company as a big chemical, vat, which is you have new people coming in, you have people leaving, you have people changing jobs. And so if you think about how do you keep the culture in a company is actually a big challenge. Because so the VAT is expanding. 800 people before COVID, 20 at the beginning, and 5,000 people, 500 today. And so if you think about the challenge we have today, I'm doing the math, 25% of a company has less than 18 months of tenure. Because you've just been growing like crazy. So think about the culture dilution. In the early days, as I like to joke with people, it was easy to know the culture because you'll be with Stephen or I every day in many meetings. We got 20 people. We'll have lunch together. We'll have beer after work together. So carrying the culture, coaching on the culture was a real-time thing that was not a lot of effort from our part. But now I have to plan things. I have to plan breakfast with employees. I have to plan trips to go to subsidiaries because when you start to expand globally, it's not like going down the stairs one floor. It's getting on an airplane and being with people that have even a different culture, how they grew up, how they were educated. A lot of countries that don't speak the language. So it's, my English, their understanding of English and so on. So it's just yet another challenge. So I think today the risk is really around execution, risk and around how do we keep the culture. Thanks. It's a great question.
Excellent. Let's see hands for questions. The light is sort of blocking my view. I see we have one back here.
Hi.
Hi.
Yeah, thanks for the talk.
Thank you.
I was wondering if you had any, you said at the beginning, the idea was sort of crazy. And so do you have any advice on knowing when an idea is sort of developed enough to take commercial, but when it's something so new and innovative like that? And then maybe related, you said the key was not necessarily being the smartest, but learning the fastest. And so what strategies do you have for learning so quickly?
Sure. Thank you for the question. So I think the thing for us was how this could change the world in such a profound way. I think you want to take a big risk because of so many unknown and the time it will take and the execution and the financing and all the things we talked about. But the price for humanity of mRNA becoming a new drug modality next to small molecule and large molecule was for us the reward. Because if we could find a path to climb this mountain, to get to one, then a lot will happen. You'll change the life of, millions, or now we know hundreds of millions of people forever. And the mental model we had a little bit was kind of the biotech industry through the Genentech and Amgen in the early days, which is look at what it has happened in the last 50 years. Think about the number of amazing medicines that have come from that field of recombinant technology. From small companies, from large companies selling companies they bought from small companies. But think about the human impact. And so that's a piece that I think will be an important criteria in your first question, which is what type of risk are you willing to take given how crazy the idea is? I think it depends on what you can do with it. And some technology have very cool one or two applications, and you have a hard time thinking about a lot. And so is it worth taking the risk? Because if it's so kind of risky from a scientific standpoint and the business and so on, but for doing maybe one or two little things, might not be worth it. But the piece for us that was really helpful, and Nubar framed it in a nice way for me, is like, will you take 5% chance to build the next Genentech or 50% chance to build the next average biotech company? And I think I can take a 5% chance to build the next Genentech. You remember that too? We both have good memories. And so I think it goes back to this upside. What is the upside for the impact of your work? And then about learning, I think you need to set a culture where it's super encouraged to ask questions. We ask why all the time. Again, the engineering training is such an amazing training. It's why, like Pareto analysis in your head, the fish analysis, and just keep going to the core science so that we can make decisions based on calculated risk of what do we know, what do we not know, what could we fail, how would we fail, how do we recover. And that's the piece that you want to create. And again, the culture I talked about with Stephen with celebrating failures. Negative information is very powerful if you play the long game. Like it's funny, the last rocket ship, Musk put in the air, not the one I think that exploded a couple of weeks ago, but I think this summer. And the media was like, oh, they failed again and so on. They must have learned so much. That's been the whole SpaceX strategy from day one. Just throw a lot of things in the air, learn every time, they're going to explode. But if your knowledge go like this, at some point, nobody can catch up with you. But you need to have that engineering scientific conviction that it's all about the learning and that you're right directionally, but you have to be humble and take a lot of turns based on the facts and the data. So I think if you need to build, and in a lot of companies, it's really hard because you have to raise money. One of the things I think we did pretty well is I was always obsessed about having several years of cash because we had a big risk with the financial markets. When the financial market crashes, like we've seen in biotech public market the last year or two, the private market is really, really hard. Really, really hard. And two is failure. At some stage, our cycle was with we had several big falls scientifically. And I remember one stage we were trying to work on liver rare disease, and it looked great in mice. And then in monkey, the mRNA went through the delivery system to another cell type, which it was not going to work from a biology standpoint. And that was a big surprise. That was a big setback. But the good news is because we had a lot of runway in terms of funding the company, the team was able to pivot without stressing, without cutting corner, because the harder the science, the more you need to be disciplined about doing the right experiment and doing all the experiments you need so you can learn and recover very strong from that failure of negative information. So you need to have a culture that understands where you're going, that is very deep into science and engineering mindset, because at some time it's going to feel really, really unpleasant. And you're going to feel the temperature boiling in everybody. And that's why you need to stay very, very cool and look at the science and make sure you have the right teams that can execute and get us to the other side. Because when you go back in your side, you rebound so fast because you learn so much. You learn things that you didn't know. And what is interesting about Moderna, if you go back into some of our historic slides, we didn't talk about vaccine for the first 2 years. But actually, one of the negative information we got led Stephen a month after with new data to walk in my office and to say, if we connect this and this and that, I think we might have one of the best vaccine technology that has ever happened. And they went to run the experiment, and he was right, and the data looked fantastic, and then we started working on vaccine. And so you need to be so humble about the science and to follow the data and to be able to work through failures. Thank you.
We have one last question from...
Thank you, Stéphane.
Yes, a joker.
In terms of learning, you spoke very, very eloquently about the internal learning within the company, but you're also working in a field that is new to everybody else, regulators, investors. Could you speak about how you helped them learn as fast as you?
Yes, it's a great question. So one of the things I always remind the teams is that all our stakeholders, the regulators, the investors, are people. And you need to build trust. Because very few people trust people they don't know, or they have not spent time with, or they don't know how they think. And so, for example, with the FDA, we went down to FDA early, not too early, because we went to Germany first. Because I wanted to go see FDA with human data. So we went to Germany first because we know the German regulators is very scientific driven. Even if it's a new technology that does weird things, as long as you understand your technology and you can prove to them you've done all the right things, they will let you do a clinical experiment. And so we went to the FDA with human data, but then we went with a lot of seminars with FDA that were not drug related. Most companies I'm aware of, they interact with FDA on a drug. But we wanted to build the scientific credibility, to take them very deep on the science. So we did several seminars. That would be multi-hour seminars, not on a drug, but just to explain the science, where every time they will ask a question that we don't know the answer, we'll tell them we don't know. And this is what we're doing to learn that, because we agree with this is really important. But today we don't know. And so it was the same with investors. They're spending a lot of time. Usually I used to joke with my CFO that every time we would raise money, that we had 2 or 3 days vacation just to do other things in the office before we get on the road again. Because a lot of CEOs, I think, are a bit naive to think you get to raise money just before you need the money. And because, again, of trust you need to set up, and people want to see, do you execute what you told me you're going to execute? And the best thing you have is time between 2 financing. And so we'll be on the road, both keeping informed of the good and the bad existing investors, but then getting to talk to new investors to explain the story, to explain the plan, and to let time get us some scores on the board. So by the time we need to raise money, they will have seen. And we played a very long game. There are some investors, I most probably failed miserably because it took me 3 or 4 financing rounds before they started investing. But because they had very deep pockets, we believe those investors were worth for a while, and we invested the time, because I was like, one day you're going to own this stock. And I made the analogy with Apple. I said, look, everybody owns Apple. And if they don't directly, it's through a mutual fund. You're going to need to own Moderna. So whether it's when we are 10 people or 50 people or 100 people or 500 people, you're going to need to own Moderna. So the only question is at what price you're going to buy the stock. Because this company is going to change medicine. This company is going to be, over time, the largest biopharmaceutical company on the planet because of its information platform where most of our drugs are going to work. Our return on investment or R&D is going to be 9x better than industry. So you don't have to compound many, many years. At some stage, you just have a velocity. If you can get on I mean, this year we're investing $4.5 billion of R&D. If we can get 90% of that money delivering drugs versus going into the Charles River in terms of returns, over time that should help us a lot. So just spending time with people, all stakeholders, and just building trust.
Excellent. Our final question.
Okay, well, I thought I would just say a couple of words, which I've never done in the 15 years we've done this lecture, because it's a pretty unusual and special occasion. And I want to say something to the students, and then I have an actual question for you, Stéphane, at the end. So 40 years ago, I came here as a graduate student. Charlie was on the faculty then as well. And as a biochemical engineering student, and I sat in this room, which is where we used to take our exams and listen to lectures. About in December, I remember very clearly, there was a set of seminars, Friday afternoons like this, where back then Ray Badur, who was the chair of the department, used to invite people from industry to come and give talks. And over the years, over the semester, I had seen the CEO of Shell, the CEO of BP, the CEO of banks. I forget if it was JP Morgan. One of the banks was run by a chemical engineer. You know, doctors, very famous medical doctors, one after the other after the other. And I used to sit in your seats and I used to say, one, what does this have to do with me? And two, could I, as a chemical engineer, do that? Like, how is it that these people did that? And I always thought that as a chemical engineer, we're trained as caterpillars, even though we're going to become butterflies. And what we end up doing with our chemical engineering is often extremely different than what it is. And it's through people like Stéphane, as I sat here listening to this, and Stéphane, I'll commend you. Stéphane knows I don't give out compliments very well, nor does he, and we're very critical. This was really a brilliant, brilliant lecture. Brilliant. I hope that you took 10% of the wisdom that he imparted, because it's several $billion of failed experiments that have taught him these things that you get for free, or just by being at MIT. It's true, that you get by being here. Your challenge is to actually look at him as an example of what you can be as a chemical engineer. And when you're trying to figure out if it's worth being what he did as a chemical engineer, understand that the science and the technology that his company developed saved over 1.5 million lives. Demonstrably, this technology, an act of biochemical engineering, actually did that. So if you can come up with a way to do even more power to you. Climate change may be one thing, but you should sit here saying, how can I do what I know how to do, instead of, I can't do this stuff, I only know how to do X or Y, but how can I do just about anything? And so that brings me to my question, which is, and by the way, this is stuff that I'm channeling Danny Wang's teachings and Charlie's teachings, because they used to encourage us to think in this way. And I'm sure the current professors do the same. So that's the burden on you. When you have been infected by what he showed you, you now have a whole life to figure out how you can be standing here 20, 30, 40 years from now, doing the same for the next generation. And so the question that I want to ask you, though, Stéphane, is how, when you look back at your chemical engineering education and all the things you've ended up doing, what of those things can you trace back to things that you took out of that, whether it's mindsets or tools or theorems or what have you, that made you able to do all of these things?
Jeez. So I'm going to hit, I'm still on here. I'm going to hit a few. I talked about a company and culture as a big vat. That's a good example of fluid mechanics, where you have things coming in, things coming out, things turning and changing in reaction. So that's a good example. I don't think there's a lot of CEOs who think as the company from a culture standpoint as a fluid mechanics problem. So that's one. I think the system thinking is another one that is fundamental. I have this mental model in my head that my job at Moderna is to keep it in balance. And I try every day to put things back in balance. This company, like any company, is a massive, complex system with negative, positive feedback loops, non-linear everywhere. And my job as CEO to do the best job I can to build the best version of Moderna I can is to permanently figure out where things are out of balance, where things are not in sync, and help the team put them back in sync. And set that culture, which again is a culture of chemical engineers, which is very complex systems that are moving all the time with a lot of variables. And that's in my head. Like, I don't have a computer yet. Maybe one day I will have an AI system helping me, or just talk to it and help me do it, but today it's in my head. And that model is really important. Another piece is how do you use time? Again, chemical reactions. I always try to push forward Moderna. I have this dynamic model in my head of Moderna today, Moderna 2 years from now, and all the piece back and forth. And my brain all the times just go backward and forward and backward and forward. One of the tools we love at Moderna is playing the movie backward, which is invent the future state 5, 10 years from now, and what needs to be true year 9, year 8, year 7, tack, 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 so that you have a chance to get there. And with all those multi-system components, that's another one. I mean, looking at the root cause of a problem is yet another one. I'm still amazed in business how some people just don't know how to really go to the true, deep root cause of a problem. And they find something, they do a little patchy thing on the first degree of root cause, and then they move on, and then it breaks again. And like, oh, it's breaking again. Like, yeah. So there's just so many things that I think this is, I mean, I'm very biased because I only know the chemical engineering training, and I don't have a PhD, so I'm not as smart as you guys are. I only have a master's. But the piece that I think is amazing about this training, that I don't know a lot of training, even in the field of engineering, is to get all those disciplines together to help train our brain to think about the world try to model a very complex world in an imperfect way, because that's what we have to do to build models to run things. And so I think that's another piece that we are all blessed as chemical engineers, is that we pick, some of us by accident, some of us by good coaching or good intuition, this field versus other field of engineering and other fields outside of the engineering world that really, I think, prepares us extremely well for the reality of this world. Whatever you do in terms of industry, you're going to have to deal with extremely complex problems, and your ability to model them in your brain, to make sure you have people on point, to make sure that you go through very deep root cause analysis of problems. Because I like to tell people, sometimes people complain they have a lot of problems to fix. And I like to remind them that if there is no problem for them to fix, I don't need them for having a job. Because I always use the extreme modeling. I say, if everything was with no problem, Nubar can run the company from his computer at Flagship by himself. And the reason we're all here is he can do that. So we're all here. And likewise. And so I think this ability to really help people see the world the way you are trying to see it. Because one of the biggest things I failed at miserably in my first few jobs is I thought everybody thought like me. Why? Because I grew up like you guys, with my buddies and my friends thinking the same, my professors. And so I was in this micro ecosystem that is not the world. And so the mistakes I made earlier in my career was assuming somebody, I said, please fix that. They will fix it like a chemical engineer. And then to realize 6 months after, geez, there's another problem, but it's the same thing. You didn't fix it. So there's a lot of things that I think as you go into the world after you graduate, whatever you do, that you have to always think about is that we're all massively biased. And your education is an incredible bias. Incredible bias, especially in the earlier years of your career, because that's most of your adult life is your education. And so how do you leverage that positively, and how do you surround yourself with people and help people get some of those tools? Because a lot of tools we learn, they're also teachable to people that have different skills that you don't have. So there's a very interesting thing about this education that we have that I think is extremely well suited for the business world in general, because it's a lot of very complex multi-systems problem. Thank you.
That's
Thank you.
excellent. Thank you so much, Stéphane. That was just amazing. We really appreciated it. And we're going to give you just one last round of applause as we complete the talk. Thank you.